So the last video I left off on this slide with the question of what happens to lactate and pyruvate once they enter the mitochondria. You can see that they both will eventually form this intermediate called acetyl-CoA, and that enters into a subsequent metabolic pathway to be broken down. This metabolic pathway is called the Krebs cycle. You can see the pyruvate forms acetyl-CoA, so this is inside the mitochondria. With that pyruvate dehydrogenase reaction, you get this cycle that's called the Krebs cycle. There's other names for it, including the tricarboxylic acid cycle and the citric acid cycle. So that all refers to the same metabolic pathway. You get a breakdown of the acetyl-CoA in this pathway. And some important aspects here, some important steps, is that you get formation of these carriers of hydrogens. So this NAD comes in, and it's a molecule that takes a hydrogen off of pyruvate. It actually takes two hydrogens off, so this should be NADH2. And throughout this cycle, you get formation of NADH2 at different points. And then you also have formation of this FADH2 as well. So you get stripping of hydrogens from the pyruvate and acetyl-CoA as it goes through this cycle by these metabolites NAD and FAD. And that's important because these hydrogens can then be delivered to a subsequent metabolic pathway called the electron transport chain, where they can be converted to energy. So they can be converted to ATP. With the Krebs cycle, you get one spot in the Krebs cycle where you get formation of ATP. And you can see that you also get release of carbon dioxide in the Krebs cycle and in that pyruvate dehydrogenase reaction. And that's the carbon dioxide that you breathe off while you're exercising. So that carbon dioxide will leave the muscle, go into the blood, and then eventually appear at your lungs, and then you exhale it while you're exercising. So you can see that with pyruvate, it's a 3-carbon molecule, and then you get the CO2 stripped off. So acetyl-CoA is a 2-carbon molecule, and then you lose those last 2 carbons in this cycle. And what I've got for you here is a quick little video summary of the entire thing. And I kind of like this one because it gives a nice little description of going from glycolysis right through the Krebs cycle.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized, and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
Okay, so that's a video summary of what happens with the Krebs cycle. So a few key things there, and there's the link, is that you start off with the one glucose molecule and it forms two pyruvates. That will in turn form two acetyl-CoAs. So the Krebs cycle goes around twice for every glucose molecule that's broken down. And that's represented by this little memory aid, the Krebs cyclers, is you actually have 2 rounds of the Krebs cycle for every glucose molecule that gets broken down. Because you have 2 pyruvates that are formed from breakdown of one molecule of glucose. This NAD and FAD, they stand for nicotinamide adenine dinucleotide and flavin adenine dinucleotide. They're derived from your B vitamins in your diet. They accept and donate pairs of hydrogen. These hydrogens act as temporary storage forms of energy. An enzyme called a dehydrogenase is involved in these reactions, where you take hydrogen off of what was originally the acetyl-CoA and deliver it to the NAD or FAD. So 2 enzyme markers of the Krebs cycle. The Krebs cycle has many enzymes, but these are the enzymes that are most commonly measured in a lab. So it's succinate dehydrogenase and citrate synthase. And they're often used as an indicator of mitochondrial volume, or the amount of mitochondria you have in a muscle, because these enzymes are only located in the mitochondria. So the purpose of the Krebs cycle, this is a bit of a summary. It breaks down pyruvate into smaller molecules. Acetyl-CoA acts as the entry point of pyruvate from carbohydrate metabolism, but it also acts as an entry point for fats and protein. So it's kind of like an intermediate in your oxidative metabolism that helps to break down all the substrates that you would consume from your diet. Produces NADH2 and FADH2. These act as temporary storage forms of energy, and it produces ATP at one step. So this is kind of like a meme here about why the Krebs cycle might be useful to know. It informs you where carbon dioxide is produced that you are breathing out during exercise. It informs you how much energy can be generated from carbohydrate and fat metabolism, so how many ATPs you can produce. And if you get into the scientific literature and look up journal articles, and they mention enzymes that are measured from the Krebs cycle, then you can understand that these reflect increases in aerobic fitness if these enzymes are increasing during a training program. And in the next section, I'm going to go over what happens to NADH2 and FADH2 that are formed in the Krebs cycle.